what data you need to collect when you collect it for evaluating your engineering prototype. And similarly, for test your hypothesis. And there's some differences in that, and we'll go through that. If you have questions while I'm talking, put them in the chat, and we'll take a break and answer them all at once, or when we're done with the slides. So the first thing is to look at the science fair has a template. There's a template for engineering projects and computer science projects. They're combined this year. And I'll give you links for that at the end. And what is specific about this, there are 12 slides. Each slide will, if you haven't looked, you need to look. That's a good thing to start there. The instructions, what to put on each of those slides. And the instructions for slide 7 is the results of the initial testing for your first prototype. And then you'll talk about you use the data, those results, to change the prototype, make a second prototype. And then you have the results for that second prototype. It's possible that if you plan things, you could have a third revision, depending how your projects go. And you could probably use slide 9 to show the results for prototype 2 and prototype 3. You can also go into slide like that. A different way of looking at this is to look at the steps. And if you look at the link at the bottom, this goes to a Science Buddies link. And what's nice there is you can click on the individual things and it'll give you more information. So you can sort of, lines up your science and engineering project the same way that the template does. Those are in a specific order to help you tell your story and present it to the judges. The difference about the engineering project is you noted that you have a prototype 1, a prototype 2, the possibility of prototype 3. And this inner section shows you that you can loop back and forth. So you brainstorm for the first one, get your data and evaluate it, test it, look at the results, go back up here and decide how to improve your prototype. And now you have prototype 2 and you collect that data and the same thing. So how do you go this? I have made up a silly little example, is thinking how to make a better water filter. And slide 1, you're going to say why you want to make a better water filter. And then slide 4, you start talking about what you're going to do with that prototype. And in that slide, you're talking about your criteria. So we said better. Better suggests that there's a standard. And then better also, better because it's faster, better because the water is more clear, or better because it gets the contamination out. And then we have to talk about the current standard. So this particular thing is going to have a prototype. When you test that prototype side by side, you'll be testing the standard. In slide 5, what are the materials that you need to create your prototype? And in this case, you also have to have your standard. So if you're looking at clarity, you're going to input dirty water, murky water, and need to measure that, perhaps in terms of sediment or parts per million. If you've contaminated the water, you're using contaminated water, then you actually need to have criteria for how you're going to measure water purity. And then depending on how your filter is going to work, let's think of it that you have a big container of input water and you're going to put it fill that volume. And then you're going to take measurements over a time period to see how long it takes for the water to become clear. And then you need to describe the equipment that's needed for the standard and your prototype. And then later, when you modify your prototype, you'll need to actually add a description of what you've modified. So then the procedures of how you're going to use it. You're going to input the water, going to use a stopwatch, how often you're going to collect the samples, and where you're going to record it. There we go. We've got data. And what are you going to measure? If it's faster, then you're going to measure something at time to become clear. Your output measure is pretty much going to depend upon, what are the current, who's bragging about what the current one will do. So your prototype has to be better than that, which means that you have to measure what the standard does. And then you describe how you evaluate and you modify it for testing. So here we have a data sheet. And we'll go into how you do that later. So you're going to record something. This is your independent variable. So you're going to have a line for the standard, all the measurements you make of the standard. You're going to put the volume. You're going to put is this the murky or is this contaminated? So you're going to have to label that. That's another independent variable. The volume needs to be constant across. You need the same volume for the same murky, same volume for the standard, for the prototype 1. Need to record the date and the time if you're going to do this every 5 minutes or every 10 minutes. I've added a column for pH just because I've learned working with science fair projects and engineering projects. If you're dealing with a liquid, it's often important to see that the pH is the same. If you're dealing with people, and this is person 1, what you want for pH is probably their age, so that you kind of hoping to control the same environment for each of them. And then the dependent variable could be output clarity. So now you're looking at sediment in parts per million. I should change that. Or purity in some units. However, this is going to involve taking samples and testing them. So if you had, you'd have a sheet of these for the standard, and as many times as you recorded it, a new set of sheets for your prototype. And now you're going to make a graph. So slide 7 this is the instructions. It says include a chart graph of your raw data. These are the results. Label the graphs and use appropriate statistics. And we'll go into that in a minute. If you want to see, here's a link to a video that talks about what should go in a graph. So I made this data up, and the 0 is the standard and X is prototype 1. And I'm looking at a measure of sediment parts per million. I didn't mark this. I didn't mark the time because it probably depends on what are the criteria that you need to match for the standard. That's going to sort of depend on this. But what's happened is whatever the time is, it's along about here, the standard plateaued and above. But your prototype plateaued, and it plateaued above totally clear. So this kind of gives you an idea of what you would do for prototype 2 is like, oh dear, my filtering got clogged up. You have to check that out. I need to have a better filter. But it's nice to know that it plateaued better than the standard. Now, appropriate statistics. Let me give you a real clue that the best statistic when you've got one observation for the standard at time 1 and for the prototype at 1, you're looking at, this is a statistic, and this is about all you can do, because you don't have 10 measurements here and 10 measurements here. So in an engineering project at the middle school and high school level, you've collected your data and you've made a graph like this. So the final thing, you can download the slides and see things that you need to pay attention to when you're making a data sheet. And you pick, you have your independent variable on every row. You have a dependent variable that you measure and the time that you measured it. You put numbers in the cells because you're going to probably want to read your data into a computer. The same number of decimal places. And it'd be useful if you have a picture of your data sheet in your lab notebook on SurveyMonkey. Now, one of the things that we can talk about in your projects, either a science fair project or an engineering project, is how are you going to record the data? You could have a handwritten table in your lab notebook and take your picture of that and put that into SurveyMonkey with your lab notebook. If you're actually doing something on the computer, getting responses from people, then you want to actually create a file on your computer that is storing the same sort of thing, so that you have, every time you take a measurement, is it for the standard or the prototype, or is it for subject 1 or subject 2 or subject 3 or subject 4, and all of this information. So you're repeating the same date, but the computer doesn't like blank cells. It's just easier when you actually get ready to make this graph. Having this sheet follow these makes it very, very easy. Okay, I'm going to stop here and get questions. I haven't looked at the chat. Are there questions? There are questions.
Yeah. Hi, Madeline.
Hi, Laura. Do you have a question?
Just a general question that everybody might like to hear the answer to from you. What you said, you want students to use the same number of decimal places in I guess in a data sheet. Can you explain that? Because sometimes it's a matter of accuracy and kids put in too many decimal places. But in this case, you're saying that you want the same number. So regardless of the data,
No,
clarify that.
Not the same number across projects, but within your data. So if you're going to measure something to 2 decimal places, you want to measure the pH to the same number of decimal places across it. So, but if I do a project and you do a project, the criteria for your project is going to determine how many decimal places to use. Does that answer your question? You're muted, Morris.
So you're really talking about just the individual variable that you're measuring, like pH. All of those numbers should have the same degree of accuracy because you've collected them all in the same way, presumably, right? That's
right.
Okay.
Yeah. So within a variable, within a column. Sanjini, your question about the survey platform?
No, we were trying to answer a student question about what is SurveyMonkey.
Yeah, I think I just replied and it just automatically went to another student's. I got messed up, but yeah, I was just trying to answer the question about the SurveyMonkey because somebody asked about SurveyMonkey.
Yeah, so yeah, that's shorthand for where you will submit the PDF that has your 12 slides and your copies of your lab notebook and the video. Let me do one quick thing. Register your project. And so if you download the slides, the next to the last slide has some links. And the sooner you start that paperwork, the better. And you will know what SurveyMonkey is at the end for that part. But just think of you're going to submit your 12 slides and you're going to submit copies of your lab notebook. And from the point of view of data collection, you should have a copy of your data sheet in the lab notebook that you submit with that. And at the same time, tossing random facts out here is that if you have results, they should go in your slides. There's some Science Buddies things. There's a video that give you ideas, and there's a sample thing. It's Science Buddies. You can actually meet with them one-on-one, emails back and forth. So download those slides. Okay, next question.
There was a question in the chat asking about our lab notebooks provided.
No, but you can use any notebook. The Science Buddies has some examples. You can go to Science Buddies and search for how to make a lab notebook. Your lab notebook should have entries from when you first start brainstorming about your project. And it'll kind of particularly recording your background research, because that's where you're going to learn how the standard water filter, if you use my example, how was it evaluated? Those units, how you made that, how do you measure pH? There's different ways to measure pH. You want to, if you're comparing your prototype to something, then you want to actually you want to be talking the same language that your standard was evaluated in. So somebody asks, is a lab notebook digital? No, it doesn't have to be. It could be. You could set up a spreadsheet in Excel or Google Sheets or LibreOffice. But typically, I think you start with actually a binder and make entries daily, what you're doing on your project. The data sheet might be easiest done depending on your comfort level with Excel or what spreadsheet you use or Google Sheets. At some point, if you're going to want to make a graph, you can take what you've written in your lab notebook and enter that digitally into an Excel sheet or a spreadsheet. In Google Sheets, you can get a reasonable graph, or better yet, you can probably come back in October and November and ask the statisticians for help analyzing your results. That's just a shameless promotion. Does that help you, Fam? Elena? I don't see you saying yes or no.
Madeline, can I just say something about lab notebooks?
Yes.
Yeah, these students will have an opportunity to post five slides that are representative of their lab notebooks. And so, as Madeline indicated, I think they're best if they are like a ship's log. They are representative of what you did on a daily basis, not really a summary of your research plan and things like that. They are for the judges to see how you recorded data that day, what kind of problems you had that day, how you thought about your project that day. So that is a real lab notebook. A digital version of that we've accepted, but in reality, if you didn't sit there with a laptop and make notes on a laptop, then it's really not a lab notebook. It can be digital, but it should be a daily log. Okay.
No, I see people nodding their heads. Maureen,
I just wanted to say that your notebook doesn't have to be super neat, but you shouldn't be erasing anything. If you take some data and then something happens, or you go, oh, whoops, I wrote the wrong thing, don't erase it, cross it off, put the right thing above it. That's what the scientists are supposed to do. And make sure you put your date and even the time on every entry on the top of the page. And that will demonstrate to the scientist and the judges that you're a real scientist. You're really doing it correctly. So if you get grass stains or the page is torn, that's okay. Don't worry about it. It's authentic and real, and it's your genuine data. So that's what we want to see.
At a face-to-face fair, we would actually, if any of you have participated in the past, we would have a lab note. The lab notebook would be sitting there on the table, the whole lab notebook, and the judges would thumb through it and see how the student did his project. So this is a virtual way of getting a feeling for what your actual lab notebook look like.
Right. So when you make 5 copies of different pages from your lab book, you want to pick the best 5 that reflect your project the best. And if that's where I said, if that happened to be scribbles on it or something else, but it's got important data and it's one of your 5 important sheets, turn it in. You don't have to worry about super neatness there.
so let me just remind you, the point of this Zoom session is that a key part of your lab notebook is actually the data that you recorded the way I showed it. And if you download the slides, you can see that that template for the data. And you need to be doing that right there as you're starting your materials, your procedures, setting up your data sheet, and then recording that. And if you're doing something on your project, observing data every day for 2 weeks, you're going to have an entry in that data sheet for each time you're doing those measurements. And if you're looking at, say you've got 10 plants you're treated with water, and 10 plants you're treated with Gatorade, you've got 20 rows in your data sheet to enter that data. And that is part of your lab notebook.
Madeline,
David, you have a project.
well, maybe this question got answered. How many trials should an experiment have? Did that question get answered?
No, because that's going to
I'd like
depend
to-
upon the
I'd like
project.
to make a comment. In some experiments, it's very hard to generate data. In others, it's fairly easy. So you report what you've got and work with what you've got. I think that's the general answer. Main thing is you have to say what your sample size is. So
The judges would be impressed. In a science fair project, if you're dealing with potted plants, number 1 is your time, number 2 is the cost, but 10 is a nice round number. It's not likely that you're going to have the time and equipment resources to actually satisfy what statisticians call to do a power calculation to be able to tell something. But the purpose of your science fair and your engineering project is to see what you can do to answer the question with the resources that you have. In a science project, you really should strive for 5 or 10 observations for each independent setup. David, does that mesh with your.
I'm trying to answer
suggest-
Kayla Lee's question here.
Go ahead, Kayla.
How many trials should you have to get an accurate piece of data? And I think you sort of talked about that, but my thought is there's no simple answer to that.
Actually, my view of multiple observations of the same setup is you're actually wanting to estimate the variability. So if you have one observation, you don't know where that is in the variability. If you take two, you at least can see how wide that range could be. And if you take three, which is it used to be that the standard middle school project used 3 observations, so you could see the median.
But like if you're doing human subjects work and doing surveys or something, you could have a much larger sample size.
Yeah.
And because humans are even more variable, again, you've got things like age and weight and education level that to control for more variability than your prototype water filter is going to be.
Right. I would also make a comment about that especially for new students. Certainly you should not, you should avoid an N of 1 or in which you are the subject, etc. That only appears to be a pilot study to see if you can do the experiment. So I think Madeline gives pretty good advice for most projects. You should seek to do at least 5 or 6, an N of 5 or 6, so you can test your experimental apparatus, so you can test your experiment to see if you can collect data that is similar, because you may end up only doing averages and means, and you may not do further statistical analysis, but you won't see very much unless you do a few repetitions with different subjects or different trials of the same variable. So that is science, repeating the experiment and seeing what you get. I think judges, regardless of what you get, will appreciate that you have tested your experiment by doing multiple N. Does that help?
Again, I would point out that the best way to present your data is actually to make a graph. So you're actually showing the raw observation, so you can see how they spread apart.